So welcome back, everyone. I hope you enjoyed yesterday's session. So this is the morning session. There's also going to be an afternoon session. This is the Data Plus Room 1 of the morning session. We're going to hear 4 posters, one on disease, emergence, and richness in primates. Another when Black stories go black. Another when on a boundary update tool for utility services, and another on human activity recognition. So there's going to be about the teams are going to take the stage to present their posters. We'll have a discussion, and then there'll be some transition time when the second poster session will start at 11. So welcome the team on primates. So whenever you're ready, feel free to take it away.
Okay. So, thank you for coming to see our poster for our Data Plus project. We have been diving into understanding the drivers of parasite richness in primates. So in this context, parasites can refer to traditional parasites such as mosquitoes or Hellman's parasitic worms, but we also expand that into viruses, bacteria, and fungi. So pretty much anything that takes energy from the host. So what we have shown here is just a simple example of a bipartite network showing a few example host primates with some of their parasites. So our project seeks to understand the factors that contribute to the actual number of different species of parasites in or on a host. We call this parasite richness. So we are studying this because this information on the drivers of parasite richness, whether geographical factors or behavioral factors, these have the potential to inform parasitology and emerging disease epidemiology, in particular, with particular relevance to humans when we're studying non-human primates.
So to conduct those analyses, we get our data mainly from the Global Primate Parasite Database and also from other related literatures. We also include phylogenetic-based predictions in our methods, so we obtained a consensus chronogram from the 10K tree website. Phylogeny could be used to show the evolutionary relationship between species. The phylogeny map shows on the lower left is color-coded by the citation count. We can see that the species colored by red, like the large apes, are studied much more than the other species, which is color-coded by blue. We need to figure out a way to control for the sampling effort.
As there are many host-parasite interactions that remain undocumented in the database, because we can't go out and study all the primates in the world, we used evolutionary relationships encoded as distances on a phylogenetic tree to predict undocumented interactions in the network. So in our model, the probability of a missing link is driven by the sum of the undocumented hosts' phylogenetic distances to the documented hosts of that parasite, which means that the model allocates higher probabilities when a closely related primate interacts with a parasite or when many distantly related primates interact with that parasite. So these two graphs show the degree centrality, which is the number of edges connected to a node, against the proportion of data. After increasing undocumented links, you can see that the average degree centrality increases for the nodes in our network.
In order to test our hypotheses about which primate traits had a large influence on parasitism, we ran Pgls, which are a type of regression that takes phylogeny of our data into account. We predicted the total parasite count for both our original dataset and also a new dataset with added relationships from our network imputation. We also included log citation as a weight and a covariate in our models to control for the range of sampling efforts in our data. After that, we ranked our models by their AIC values and also obtained coefficients through model averaging. We found that for our original and imputed datasets that diel activity, which is when an organism is active, locomotion, whether a species lives in trees or on the ground or both, mating system, and realm were the most important predictors of parasitism. And all of us would like to take a moment to thank Marie and Charlie for being amazing project leaders this summer, and also to thank all the people who have given us so much help and advice throughout. And also a big thank you to everyone running Data Plus and everyone for attending our presentation. We're now going to open the floor for questions.
Thanks a lot for the introduction to your poster. Does anyone have any questions that they've gathered from the poster so far, or anything that they want to sort of steer the conversation towards? So one thing I was wondering, could you go into the Pgls analysis and maybe zoom in a little bit on what's happening in the y-axis and what those differentiators are?
A little bit zoomed in. Yeah, so the Pgls analysis, it uses a linear approach, but also takes into account the phylogenetic distance between different species. So one really important thing to take into account when you're studying parasites and traits between related species is that the phylogenetic relationships between them has an effect on their similarity in terms of their ecological traits and their parasite sharing. So their different species are not actually independent due to phylogenetic relationship. So using a phylogenetic generalized least squares regression allows us to take into account that dependence.
Cool. So Sarah Dwyer asks, what challenges did your team face? How did you address these challenges, and what did you learn from that experience?
I can take this one. I think the biggest challenge that we consistently face throughout this entire 10 weeks is that of like controlling for sampling effort. Like we tried so many different ways, but in the end there's still like because we have to measure like parasites using like either total parasite count or like number of citations or some different metric, but all of them keep getting affected by like the poor sampling of certain species. There's just simply not enough data. So we kept trying to find ways to like level it out, but there's still a little bit of issue with that we're working on.
Yeah, compare, say, a chimpanzee or gorilla to some mouse lemur in Madagascar. So the chimpanzee is way more studied than the mouse lemur, and so therefore it's going to have way more parasites. I'll take Tad Dallas's question when we get to it.
Oh yeah, absolutely. Is there anything else you want to talk about your just experience with Data Plus, or should we move on? And Sarah, did you have any follow-up questions on that? Cool. So thanks. So Ted Dallas asks, I was curious about how many links were added to the network. From panel 3 in the poster, it seems like it's a fair number of links, which could be realistic. Also, great presentation and super interesting work.
Yeah. So so for those who are unaware, Tad Dallas actually helped us quite a bit in our network work. We looked at his code and used it. He had done previous work in network imputation, but we had found a more recent paper, Dallas, by El Masri, and what he had done was he had also looked at this problem, and part of his code was looking at. Sorry, there's some type of feedback. Part of his code also looked at predicting based on phylogeny, and then 2 also looked at how many edges do we actually have in the data, and he showed that we had about predicted we would the ones that we actually observed in the network was about 21% of the data, and so when we used his thresholds of predicted of predicted edges and put it into the data set, it amounted to close to about that we had about 21% of the data. So it was a bit different from your data. We're still going to investigate and look into and see if this this huge amount of predicted edges is actually meaning something, because again, as we discussed with sampling efforts, it does tend to predict on things like chimpanzee and yellow fever, which already have a lot of edges. It tends to start connecting those to everybody. So we definitely have quite a bit of work to do on the networks as well. But this was an interesting beginning results on these host-parasite interactions. Does that answer your question, Tad?
And another reason that we decided to go with a phylogeny-based model rather than relying on our trait data is because of how sparse our data set was. So using phylogeny helped us sidestep this issue, because phylogeny acted as like a fully connected network because we knew the phylogeny pretty well.
And actually, Jenny, could you expand on what phylogeny is?
Yeah, so in the slide with our poster, Courtney, if you could, or the phylogeny. Yeah, so in a phylogeny is basically like the relationship or the evolutionary distances between different species of primates. So it's like a phylogenetic tree.
Cool. And then I guess just to could you say one more time about just how the phylogeny relates to how the parasites interact with these, how they interact with the phylogenetic tree?
Yeah, so the way that our model predicted whether an undocumented parasite interacted with a host was based on the phylogenetic distances, or the sum of phylogenetic distances from that undocumented host to all the documented hosts of that parasite. So the more closely related, so that means like our model would allocate a higher probability of a host interacting with a parasite if closely related hosts also interact with that parasite.
Cool. So Monica asks, hi, great presentation. I love the work. Great. If your group was allowed to work on this project for longer, do you have future directions in mind?
I can take that. We definitely plan on working on this project for longer, actually. We're really excited to carry this forward in the Nunn Lab in the fall. And we do want to, I think some of the biggest future directions are to nail down some of the methodological questions that arose that we kind of had to skip over in the interest of time. And one of those is, how many imputed edges should we add into our model? Another one is, what are other variables that we could use in those imputations of edges, like what Robert said, we could use climate to possibly get even more accurate predictions. And then just looking at some parasite level variables as well that we didn't look at. We mostly focused on host traits, but we could look at parasite type and transmission mode in some of our analyses as well to see how those affect primate parasite richness.
And I guess from the point of view of doing a data science project in this, I guess there's the tension of sort of your project is going to suggest certain relationships between parasites and primates, which could then go be tested in the field or looked for in the field. To what extent do you see, to what extent do you think that this project is ready to go be sort of field tested in some ways? And what would that field testing look like in a way that you'd be excited about? And to what extent does the current analysis that you've done say, oh, if we had this analysis, or if this study was done, or if, you know, if we were able to access X, you know, what's the next set of data that you would like to see given the work that you've done so far?
I could take that again unless somebody else feels inclined. I think since there is so much variation in the number of studies on different primates and studies on different parasites, there's huge variation in parasites that have been studied as well, with viruses, for example, having lower citations. I think it would be really exciting for a lot of those less studied species to be filled in, and then the parasites that are found in those species that have been studied either not at all or maybe only once or twice, we could see whether those correspond to some of the predictions that we have made.
There's the ethics behind that as well, which is, do you look for those parasites in the wild? Because, you know, as Robert was saying, it could be a issue of climate where the species don't have access to those parasites, or the parasites and the species don't kind of overlap, or do you do this in a lab setting? And yeah, there's all sorts of very interesting ethical questions that go along with that too.
It's true, and there are a lot of studies of parasite infection in laboratory primates, but the global primate parasite database is only focused on parasites in wild primates. So I'd say that is a major limiting factor, because that's really the only method that will give more data to this type of study.
Yeah. So Tad Dallas has another question, which is, to what extent can your approach be used for prediction? Is it possible to leave a host out and then predict the parasite species richness, for example, the degree of centrality?
So I think that would be an interesting next step to do. We did a little bit of that with the El-Masri code. It already has a five-fold cross or n-fold cross validation, and based on that, it seems like the predictive accuracy is fairly accurate with an AUC of about 0.92. But yeah, that would definitely be. I'd like to dive more into the code on that with more time, and to really get into that type of prediction and how certain that is, and I'd like more work on that, yes.
And I guess I skipped it over just because it was partially addressed in one of the answers, but let's maybe go back there. So Robert Calderbank was asking, about other parasite dependencies, such as climate. And I was wondering, and I know you've touched on this, but I was wondering if you wanted to expound on that answer at all, or talk about some of the other variables that may affect what parasites affect what primates.
So we don't have a direct variable like climate, but we do have like a geographic variable like the latitude or the total geographic area. I think we can kind of indirectly reflect the climate of it, but we do not currently have the direct climate variable. We can look into that to see if we can get some kind of this variable from other literatures. But we do test the centroid latitude for the Pgls model, but it seems like it's not that important. But and also we did a log transformation for all the numerical variables, and then after we did the log transformation and all the numerical variable became like non-significant, we need to figure out a way how to like deal with those variables in our future analysis.
Cool. Are there other places that people would want to draw the discussion or other questions that people have? Oh, so Tata adds, one thing you may want to consider is geographic range size instead of climate directly or in addition to. This can be calculated by pulling species occurrence records from the GBIF and has a good bit of theory underlying why it's important.
Yeah, definitely. We calculated total geographic extent from the IUCN. We still have to, one of the things we have to do next is go back and make sure our projection for that is correct, so that, because we found that it wasn't very important in predicting parasitism, but we need to go back and ensure the quality of that variable is as it should be.
Oh, go ahead.
No, go ahead, Andrew.
And we also have, so we have the total geographic area of the species, but we also have home range as well. So how far they travel, how far each trooper species or what have you travels within that range as well. If that's also
one challenge we ran into and kind of like wanting to run our model on which traits predict parasitism, but then also wanting to make predictions, is we were trying to not use the same variables in both predictions and then later in our model, because we weren't sure about the redundancy of that. So that was another problem that we had to kind of navigate, and that we'll be navigating in the future as we add in variables to the different parts.
Yeah. Well, cool. Well, thanks a lot. Let's give this group a virtual round of applause. If there are any other questions and people want to chime in, go ahead. But I think you guys will probably stick around for another minute or two at least. And yeah, we'll take a short break